biology. This will be our video on facilitated diffusion. And we have already discussed diffusion, and facilitated diffusion is a slight modification of that. It comes from the word facile, which means easy, and so facilitated diffusion is diffusion made easier for certain particles with the use of transport proteins. We've talked about the fluid mosaic model and how transport proteins are embedded in the cell membrane. Well, these transport proteins can respond to certain circumstances and help move things across the cell membrane. So let's get started with those. They involve two different types of transport protein. Facilitated diffusion uses two different types. The first is called a channel. So we actually are familiar with channels because we discussed one already. The example of the channel that we are familiar with is aquaporin, which helps diffuse water. Well, channels are all similar in that they all have a tunnel shape. There is a clear passage for particles to pass through, and they determine what can and cannot pass through that based on the size and polarity of the particles. So, for instance, in this particular model, we have these hexagons, which can pass through this because they're the right polarity and the right size, but perhaps these red ovals are too large to pass through, so they can't. So the tunnels, and if something is the wrong polarity, it also won't pass through. So they determine if something can pass through based on size and polarity of that particle. Another thing about tunnels is because they're simply an open channel, or another thing about channels, because they're simply an open tunnel, is that they can go in either directions. So when the concentration gradient changes, the direction of the molecule's movement will change. So right now, the particles are moving from a high concentration of hexagons to a low concentration of hexagons. But should the situation in the cell change, then we might have a higher concentration of hexagons here, and we would go the other direction. So channels are two-way motion, and the motion changes based on the concentration gradient. That's it for channels, so let's go on to carriers. Carriers are a little more complicated. Carriers, unlike channels, which are non-specific, anything that's small enough and anything that has the right polarity can get through that channel. Carriers are very specific to one particle. They'll only move one type of particle, and in this example, the particle that's being moved is glucose. Another thing that is different about carriers is that they will only move their particle in one direction. And since this is diffusion, it has to be with the concentration gradient. If the concentration gradient changes, then the carrier becomes much less effective, and it's very, very unlikely to work. So it essentially turns off when the concentration gradient flips. So it has one direction, and when the concentration gradient is with that direction, everything is good. But when the concentration gradient flips, the carrier essentially stops working. So how do carriers work? They're not a tunnel. They are much more complicated. So in this diagram, they've simplified the complex protein shape. But remember, this looks nice, but they're really a wild, squiggly, multi-folded, coiled polypeptide. That's what proteins really are. So how does this happen? Well, first, their target particle, the specific particle that they move across the membrane, is attracted to the protein based on its polarity. Now, once it's there, the charges in that particle will disrupt hydrogen bonds in the protein, causing the protein to change its shape, which they're kind of simply illustrating here with these arrows. So first we attract the particle, then that causes an isomerization. Next, after the isomerization, the charges in the, what is happening? The charges in the protein have moved around a little bit as a cause of that renaturing, and now the particle is no longer it's no longer attracted to the protein, and so it is released. And once that particle is gone, there's nothing interfering with the shape or charges of the polypeptide, so it renatures back to its original condition. So they attract the particle, they isomerize, they release, and then renature. So let's review. Facilitated diffusion is diffusion made easier with the assistance of transport proteins. And we have two types. Channels, and we are kind of familiar with them because aquaporin is an example of a channel. They are all tunnel-shaped. They restrict passage of the particles based on size and polarity, and they allow for two-way motion, and they shift between them as the concentration gradient changes. Carriers, on the other hand, move a specific target particle. they only move their particle in one direction, and only when that is the same direction as the concentration gradient. To work, carriers first attract their target particle based on its polarity, size, and shape. Then, that target particle triggers isomerization. It triggers isomerization by disrupting the hydrogen bonds, and during that isomerization, the target particle is moved across the membrane. After the isomerization has occurred, the target particle is no longer attracted to the protein because the charges have moved around, and the particle is released. And finally, when that particle is gone, the protein renatures because the particle isn't able to disrupt its bonding anymore. I hope this helps. Struggle well.